Hello, I am Benjamin Miss. This is Physiological Psychology, Dreaming, and Consciousness. The first thing we'll look at here is the EEG signal. This is where a lot of our knowledge started out in terms of neuroscience, like what's actually happening, and can we detect these changes in the brain? These signals represent large numbers of neurons that are firing together. And more recent technology has allowed for the very rapid disaggregation. So we look at these top lines here. These represent the left and right hemispheric EEG. So there's a lot of signals all being combined into these very fast-paced, jaggedy lines, right? Well, they can decompose the signal and pull out the component parts rapidly, almost in real time, on any kind of basic computer, which allows you to look at the various components. So you see this wave, and this wave is made up of these other pieces. As you see down, you see alpha waves and beta waves. And so these waves add together along with all the other components and random noise and give you this final EEG signal. And it's pretty cool to be able to see the technology pulling this apart and seeing these. This is actually an example from some technology we use in the lab that students get to do EEG with. Now, the waves we tend to talk about the most are these. And the first ones are beta. Beta are kind of like your conscious, anxious, alertness waves. If you're doing a task, right, you can usually detect these, maybe in a greater way. The alpha waves are relaxation, and you should be able to see, well, EEG we talk about in terms of sleep, and when someone's asleep, we can see them going through the different stages of sleep. But when it comes to awake states, we can still see differences in the amplitudes and frequencies of the waves between awake and alert and relaxed. People who can meditate really well, guided visualization, you might even be able to detect theta waves. That's a little controversial. Delta tends to be very deep sleep. And then we've got this last type, gamma, right? So we look at the various frequencies of these waves, where beta is 12 to 30, alpha is 7 to 12, theta is 4 to 7, delta is 0.1 to 4. Now, these are all measured in hertz, and hertz is cycles per second. So it's the idea of like a sine wave, where like how many peaks are in a second. We use hertz when we measure auditory perception. We're looking at sound waves, right? The frequency is measured in hertz, and that's how close the peaks are. If we recall from auditory, close together peaks of these waves is a high-pitched sound, and long spaces between them are low-pitched sounds. And so we're looking at frequency in terms of our EEG signal, right? And people will often call these brain waves, which I don't really like that term, but it's fine. So 0.1 hertz means the wave takes 10 seconds to repeat. That's basically 6 waves per minute. Now, by waves, we're saying, what are these waves? These waves are aggregates of thousands, millions of neurons that are all firing in synchrony. So they're firing together in this, suggesting that they're working together. And in deep sleep, you get these huge slow waves, lots of neurons firing together. Awake and alert, really small, fast waves where there's a lot of noise, neurons doing different things, but they do still tie together. And we can detect these networks. Gamma waves are interesting because these are the ones that are associated more recently with consciousness, right? Coherent, unified perception. And these go from about 30 to 100 hertz, very fast. And the area they look at the most is about 40. And this is almost this mythical, magical, frequency where conscious perception is taking place, tied together through likely the thalamus, where all these various brain areas that are at this frequency are whatever you are consciously aware of. Widely accepted, controversial, but it does seem to be reasonable. And you'll notice the ranges here. There's a little bit of variability in estimates. It's the same kind of thing where depending where you look. So this is the general idea. Now, in terms of analyzing the EEG, there's three main methods. So we say three types of EEG. This is more like three types of analyzing the EEG signal. The first is looking at the signal. What does it say? So this is where the earliest work was done, where you put the electrodes on someone's head and they sleep, and you look and you can very clearly see them going through the stages of sleep. And we'll see that. Then you have the ERP and also VEP. ERP is event-related potential, right, or evoked potential, which is you have people look at a similar stimulus over and over. So for the VEP, for instance, you might show a video of a checkerboard, of a flashing kind of checkerboard. And what's happening is you can actually measure that, your optic nerve sending the signal to your occipital lobe. You can actually measure the oscillations. However, there's a lot of noise. So just measuring it one time, one flicker, let's say you're flickering it 2 times a second, which is 0.5 hertz, you would see, that'd be 2 hertz, you would see these little spikes, right? But you might not, because there's so much noise. There's so much noise in the EEG signal that it's hard to see, to tie it to an event. We want to tie your brain function to an event, and that's hard to do. So ERP and VEP, the visual evoked potential, which is specifically looking at the optic nerve function, you can actually see if you flash this checkerboard every twice a second, and then every half second you grab that output, and then you grab the next half second, average it, the next half second, average it, the next half second, average it, you eventually will see this peak where you get this switching, right, where this signal is changing and the communication is changing. So it does require some more computational technology. And then the last one is called steady state. This is the newest because it requires that high computational process of pulling out the component parts. Similar to the way we saw the alpha and beta waves pulled out of that signal, this is how that would work, where you can see the actual frequencies and say, hey, if I show someone this stimulus and I show someone this stimulus, we're actually seeing a difference in frequency, which suggests a difference in processing. Now remember, the issue with EEG is this. We don't actually know where, right? We don't actually know where in the brain things are happening, but we know things are happening. Now we look at sleep, and we're going to use EEG to study sleep, but here we're looking at what are called endogenous cycles, because sleep is endogenous, and endogenous means generated from within. And some animals produce these, not just are the typical ones that we discuss, which are circadian, but also they're circannual, which are, over the course of the year, right? And so here we see, for instance, animals that migrate, animals that hibernate, right? These things have these mechanisms that, will change. Before an animal hibernates, they bulk up, and it changes their eating habits, their sleeping habits, all these things. When we talk about, what humans produce, we do produce more than just this, but our circadian rhythms are these 24-hour cycles, right, which match up with the environment, which the light in the environment, the world around us, the day. And so these are generated, from within. And again, to a certain extent, this also regulates the frequency of eating, drinking. Your body temperature fluctuates throughout the day in very predictable ways. Your hunger, being hungry, very predictable. When you are secreting hormones, your hormone changes. And there's these other cycles too that are maybe monthly cycles that people have, and seasonal cycles. Well, with our circadian rhythms, we see some interesting patterns. For instance, if you take someone and you give them more darkness than light, you will see increased depression and reduced activity. You think of someone who lives in near one of the North or South Pole, where they have extremely long days in the summer and extremely short days in the winter. And by days, I mean the amount of sunlight. And you can absolutely see these behavioral changes. When you have constant light, you get sleep disorders. So people who work at night, who are being bathed in light at night, will have this also, right? And you get this delayed sleep phase where they have trouble falling asleep, trouble waking up. But if you have this, bright light in the, at different times, right, all day, then your sleep patterns get really out of whack. We also see, if you imagine an astronaut orbiting the Earth, they're seeing the sunrise and sunset multiple times a day. Their sleep patterns get messed up too. When we look at what happens when you actually take away this, conquering of nature that we've done, right? We've got light all the time. You take that away, and what do you get? Well, if you were to actually take people and put them into an environment that is more typical of our history, you see, for instance, that people will often get into these 2 sessions. So only 10 hours of light per day. Imagine a typical winter in much of the northern hemisphere, right? Where during the colder, darker months, your days are shorter too. And the amount of light you get is maybe 10 hours a day. Well, that means you've got these 14 hours of darkness. What are people going to do? What did they do historically? They didn't sit there and light candles and sit out and stay up late. When it got dark, they went to sleep. And when they went to sleep, they would, sleep for maybe more than 1 session. They'd sleep for 4 hours, wake up, and then go back to sleep. And sometimes during this times, people would meditate, pray, be with their loved ones. So there's different things. And, I have this image here. You look at, the power outage from Hurricane Sandy. This massive hurricane knocked out lights in lower Manhattan. It's still bright in the sky. There's so much light. And this is extremely important in terms of how we function. But, we want to maintain that. Why do so many young people and any people really have insomnia or have poor sleep habits? We need this darkness to help us to function. And there's ways to cope with that in our modern way. We'll look at that. Here's how our circadian rhythms change over time. Brand new babies have a massive amount of sleep and also a massive amount of REM. And then as we get older, you see that both of these things lower. Less sleep and lesser proportion of REM, right? This is the it's called rapid eye movement. It's one of the major components of sleep. And this tells us that you get this. But again, these are averages. You take people at different ages, you're going to get a wide variety of how much sleep they're actually getting. We look at, the purpose of this is to keep us in sync with the outside world. And our clock generates a rhythm by itself of slightly longer than 24 hours. So most of us have this kind of phase delayed, right? This might be why more people have trouble falling asleep than falling asleep easy, because you have this phase delayed where your circadian rhythm is slightly longer than 24 hours and needs some help getting reset. Normally we can get that, but there's a lot of factors that don't. Again, depending on the amount of light you get per day. Cloud cover can have an effect even. Your shift, working indoors, these things can all have interesting effects. So how do we reset our internal clock? What's the number one signal that we use in order to reset our 24-hour circadian rhythm? Well, these are called zeitgebers. Okay, things that are used to reset our clocks. And the number one that we use is sunlight. We can also use anything that's rhythmic, anything that has a schedule. So let's say you do shift work, okay, or let's say that you're indoors. What's important is maintaining a schedule, exercise, meals, whatever it is you're doing that give you this these signals. Environmental temperature. It gets cooler in the evenings usually. These signals that help us sleep. But when we don't have sunlight, this can be a problem. We look at other things like jet lag. Why is it easier for most people to travel west than it is to travel east? Well, phase delay is actually easier for most of us to cope with, especially because we have this slightly longer than 24-hour internally generated clock. So what happens is, when you travel west, the days are a little longer. It's easier to adjust to a longer day than it is to a shorter one. Traveling east, you lose that time. So let's say you're traveling from the west coast of the U.S. to the east coast, a 3-hour time difference. So you, all of a sudden, you're used to the sun setting at, let's say 7 o'clock in California, and in Connecticut, the sun's setting at, all of a sudden, the sun's already down when you get there, So you fly to the coast, it's 3 hours later, and the sun's gone, right? So you are 3 hours, and so you end up with this shorter day, so all of a sudden, it's hard to kind of adjust to that. You travel to the west coast in the other direction, let's say you use the sun setting at 7, now it's setting at 10 p.m., but that's easier to deal with because you're going to adjust more quickly to it, and your internal clock is going to go back and be like, oh yes, this is 7 o'clock setting, not 10 o'clock, because you just stay up late one night and you adjust, usually within a short period of time. Now, you also look at what happens. So people who work the night shift have more accidents. We don't seem to perfectly adjust, even if we have a lot of experience with it, to these shifts. And the reason for that is we need, this signal. It's typical for us to be up during the day, not at night. We're diurnal. We're not nocturnal. When you look at how this is functioning, we look at where. And the structure we're going to look at is called the suprachiasmatic nucleus. And this is going to control the production of what's called melatonin. And melatonin is associated with sleep. When you're tired in the evening, your melatonin levels are high. In the morning when you wake up, your melatonin levels are low. And you get this signal that tells us this. And you actually have a couple of very interesting things happening here. So, the suprachiasmatic nucleus is part of the hypothalamus. And the hypothalamus is going to be our focus for a while, because the hypothalamus deals with hormones. These are hormones for regulation, our internal body states. These are hormones for sexual behavior. And so we're going to look at this tiny area, and you can see it here, this tiny area called the hypothalamus, below the thalamus, under the thalamus, that it contains these nuclei that control all this stuff. And what's interesting is it's just a big hunk of matter, but we know there's a different nuclei by putting electrodes in there. Recent research suggests that it's the hypothalamus and the degradation of it, right? So you know how cells change as we age. The part that regulates our body is where you see the biggest issue. So because it changes, because these cells tend to break down, they don't get replaced, you see just kind of, cell death over time. This has the biggest effect on us physiologically in terms of age. This could be a big, big part of it. Well, it's located above what's called the optic chiasm. If you recall, we've got our optic nerves from our eyes crossing at the optic chiasm, and then our visual fields then go to the opposite side, thalamus, and then to the occipital lobe on that side. Well, this is going to make sense to us because we're going to look at where is the light coming from, what's that signal. And if this is damaged, then you lose that synchronization. Your body rhythms get out of sync. The suprachiasmatic nucleus generates a circadian rhythm on its own. So basically, it needs this outside information to help it, but it has this controlled kind of rhythmic signal that it sends. And the frequency, remember the frequency of neuron firing, right, is going to matter here, because the frequency is going to change based on what the signal is. So when you take it out, when you take a cell from this and you raise it by itself, it's going to produce an action potential in a rhythmic pattern. Well, here's our hypothalamus, and here's the nuclei, a whole bunch of these things. So again, we see it here in the brain, and then we see it kind of by itself. And hanging from it, right, is the pituitary. And you've got two parts of the pituitary, the anterior and the posterior. And so basically this is kind of like resting. The hypothalamus is right above kind of some bone, some of that sinus bone in the front part of our, kind of part of our skull. So it's resting there, and there's a tiny hole in it, a tiny foramen, through which the infundibulum connects the pituitary. So basically, the pituitary is directly connected to the hypothalamus, and the posterior pituitary effectively contains brain cells. In the same way we think of our eye as brain, our pituitary, a gland, part of our endocrine system, is also brain. It's connected directly. And we see here this suprachiasmatic nucleus. It's positioned above the optic chiasm. Well, what's special about this is we've learned about two paths of light to the brain. If you recall, the geniculostriate and the tectopulvinar pathways when we talked about vision and the occipital lobe, where the geniculostriate is our conscious perception of vision, and our tectopulvinar is our kind of our animal vision, which is like something draws your attention or makes you look. Now, we've also got a third pathway. This isn't as much information because what's being sent here is basically a direct signal to the hypothalamus, is there light? And so you've got a set of ganglion cells in your retina, right? You recall you've got your rods and cones, you got your bipolar cells, you got your ganglion cells, a bunch of ganglion cells that have their own photopigment. This photopigment is called melanopsin. That's convenient. It sounds like melatonin. Well, melanopsin basically says, is there light? Specifically light in the blue range of the spectrum. That shouldn't be surprising at all, because what kind of filters are built into most of your phones and computer screens? Blue light filters. These filters make your screens take on a yellowish tinge. And this is good because it's reducing the amount of blue light. Basically, whenever the sun sets, you should have your blue light filter come on automatically. You should have the lights in your home, those warm yellow lights, right, rather than the bright white, because the white has more of the blue frequency. And that blue light, that blue frequency, is what's telling your brain it's daytime. So imagine how confusing that is for your hypothalamus. Here you are going about your business, the sun sets, and there's still all this bright light. So your hypothalamus is sending the signal to your pineal gland, which produces melatonin, and saying, hang on, don't produce it. It's still daytime here. It's not time to sleep. We need to get away from these evening lights to help us regulate our sleep patterns. This is called sleep hygiene, right? Having good sleep behaviors rather than looking at, very common, looking at the phone in bed, lights off, and yet you got this bright light shining in your face. And blue light filter helps. Not doing it all is better, but the blue light filter helps. So we've got genes that are responsible called period and timeless. And these period genes are called PER proteins, and the timeless genes produce TIM proteins. And these increase activities in the suprachiasmatic nucleus. These help us to regulate these things. And these gene levels are lowest in the morning, highest in the evening, and light activates a chemical that breaks down the TIM protein. So you've got these helping to regulate us and maintain these patterns. And we see here that mutations, right, things that can basically change the PER proteins from the period gene can create problematic, odd circadian rhythms. When we look at the visual pathways, here now we're looking at our three, if we think about our three pathways, right? So what do we've got? We've got our geniculostriate and tectopulvinar, which we see on the far right here. And we've also got our pathways directly to the to the SCN, the retinohypothalamic, the RHT, retinohypothalamic, the pathway directly to the suprachiasmatic nucleus, and GHT, geniculohypothalamic. So you are getting a signal to the hypothalamus from there also. Well, the suprachiasmatic nucleus is going to regulate waking and sleeping by controlling activity levels in other parts of the brain. So the suprachiasmatic nucleus is sending a signal to the pineal gland. This is part of the endocrine system. It's located in the brain posterior to the thalamus, near the midbrain, where you actually have a little gland that's producing melatonin. And this is going to help to tell you, be sleepy. It's time to sleep. It's an important signal. Now, melatonin usually begins 2 to 3 hours before bedtime in terms of secretion, in terms of ramping it up. And why? Because usually for us, the sun sets 2 to 3 hours before bedtime, and that's when you start to produce it. So when people take melatonin supplements, and because they have trouble falling asleep, it's recommended to take them 2 to 3 hours before bedtime to help your body kind of start adapting. Quite a bit. I've read quite a bit about melatonin. There's a lot of drugs that are marketed for sleep. Melatonin is not one of them because it's extremely cheap and no company owns the patent to it. A lot of the drugs given for sleep are extremely dangerous in terms of addiction and in terms of are you actually getting real sleep. So it's important to talk to a physician who knows this stuff before you use anything. Melatonin doesn't seem particularly dangerous. It also tends to be used short term. And in the papers I've read, the doses range that they use between 0.5 milligram to about 5 milligrams, which a lot of people are responding to 2 milligrams. And they take it only for 6 weeks to 3 months, right? Maybe very few studies go on longer than that. And for a portion of the population, it can help people to regulate their sleep. And once it's regulated, your body should continue to use it. But again, what's the problem with taking things like melatonin? Well, it's not terribly well regulated, and you need to find supplements where you know exactly what's in there and that these things have been carefully controlled and measured. Due to regulation issues, it's not really strong there. So melatonin resets the biological clock. And it's not like you have this 24-hour cycle that stays on there. All these signals, like the Zeitgebers, like your melatonin, are going to give you that information. So let's say you're traveling West Coast to East Coast. One thing that people might do is take melatonin, in order to help help adjust. But it also doesn't seem to work for everybody, so it all depends. All right. Which nucleus of the hypothalamus regulates melatonin production? Is it the preoptic area, the suprachiasmatic nucleus, the lateral geniculate nucleus, or the lateral hypothalamus? And the answer is the suprachiasmatic nucleus. What's the structure that releases melatonin? The preoptic area, the suprachiasmatic nucleus, the pineal gland, or the pituitary gland? And the answer is the pineal gland. In fact, if we talk about the pineal, pretty much all we're going to associate it with is really this. I mean, it does other stuff, but this is key. When we look at sleep, right, we know that it's this state of, decreased brain activity, and we don't respond to things as well. And we'll look at these different states, including coma, vegetative state, minimally conscious state, brain death, what this means. So I mentioned here sleep hygiene again. Do you have a good process? This moderate decrease in activity, this time of rest. Do you have a good process? Do you maintain a good schedule? Well, there's some really interesting things related to these. So coma is not sleep, right? It's not just like a long, deep sleep. It's an extended period of unconsciousness with fairly steady low brain activity. And it's hard to get people out of coma because they're not responding well to stimuli. Coma can be permanent. It can also be temporary. There's a lot of factors involved, and it depends on why someone's in a coma. Oftentimes, it can be mild damage that the brain is able to repair. Sometimes it's not. Now, coma is different from a minimally conscious state. These are occasional brief periods of purposeful action with limited speech comprehension, but there's this reduced level of consciousness where people aren't really able to be kind of pulled out. Now then it gets even more interesting. So the vegetative state is alternation between, you see sleep and arousal periods, but there's no awareness of the surroundings. There may be some autonomic arousal to pain. So someone can be in a vegetative state, you jab them with a pin and they're going to pull their body part away. But there's no purposeful activity, and there's no real response to speech. The only part of the brain that's functioning is the brainstem. And this differs from brain death, where there's no sign of brain activity, no response to any stimulus. In brain death, somebody could be kept alive, right, using a ventilator and something to cause their heart to beat. But there's nothing in the brain that's doing that. In a vegetative state, someone could require a feeding tube, could be kept up, but might not need those because they could still have their brainstem. And your brainstem is responsible for things like your heart beating and breathing. So here we see Terry Schiavo's brain. And that's on the right, and on the left is a typical brain. Well, Terry Schiavo was a case from the 90s that stretched on through the 2000s. It's actually a tragic case. It's a case studied in terms of medical ethics. And it's something that as people studying neuroscience, you look at this and you're like, wow, I know way better than the politicians that use this poor woman to score political points. So there was this battle between her husband and her parents about whether she should be kept alive. Well, let's look at her brain on the right. What's missing? So much. So much. Extremely reduced amount of tissue. She had an extreme, something like a stroke, and her brain just lost that oxygen, and a whole bunch of it died. She never revived, but because she was in a vegetative state, she would show some autonomic arousal, and her parents wanted her kept alive, and her husband, wanted to allow her to peacefully die. If you look at the brain on the right, there's, very little chance that this person would ever wake up. And when they do, they certainly wouldn't be themselves. And so the question that I would think is, if this was you, if you had this much brain damage, would you want to be kept alive? Or would you want them to let you go peacefully? And I think that's the interesting question. But people were like, no, life at all costs, or you're committing murder by removing things. And many people are afraid of these kind of wrong-headed laws and involvement, where if you create your own living will document, you can have it in writing that this is what you want under these various circumstances. So for your outcomes, you can have these things. So Terry Schiavo, you had different politicians getting involved, including at one point at the federal Congress, basically saying, trying to dictate medical care, which is, again, quite problematic and is useful to study in terms of what is, what's happening in brain science and neuroscience, what's actually functioning in this brain. And I believe what ended up happening is after the case had kind of cleared, most of the population was rather outraged with the whole discussion. Locked-in syndrome is interesting because this is different from a vegetative. Here we see conscious, vegetative, minimally conscious, and locked-in controls. Someone who has locked-in syndrome could have severe damage. They could also have mild damage that simply makes it so that they can't function or respond. And there are some interesting things that have been done with locked-in syndrome in order to kind of allow them to answer yes or no to questions. But just like anything else, you can have a huge range of someone who is almost completely cognitively aware to people who have who is locked-in syndrome, right, unable to communicate, to someone who is severely damaged but is able to answer some really basic yes or no questions. Now we look at sleep, right? So we see these other states that are different than sleep, and now we consider how sleep works. So here's EEG recordings during sleep, and we see awake and then stage 1, 2, and 3. And you see 3 and then 4 here, kind of like 4 is like a bigger version of 3, right? These huge, slow, oscillating waves. And then REM, which looks a little like a combination of stage 1 and awakeness. Well, the EEG allowed researchers to discover there were various stages and compare them. And then you also have something we look at sometimes called a polysomnograph, where it's a combination of an EEG and an eye movement recording. And this allows you to see kind of that REM is happening during, rapid eye movement is happening during that stage of sleep. Now we also see here, we see our sleep stages over the course of the night. And this is interesting too. This part at the top is REM. And one of the things you should notice is that REM and your other stages are happening much more commonly during later stages of the night. So if you only get a few hours of sleep, you're getting that deep sleep. So you may be getting that body recovery, but you're not getting that, all of these natural processes. And we know that anything that has neurons has some type of sleep, which suggests it's a natural component of having a brain. When you look at this, we see that, when someone starts to fall asleep, you get these alpha waves. And in stage 1, you get this, just begun sleep, jagged, irregular, low voltage waves, similar to being awake, but brain activity is declining. In stage 2, it's characterized by the presence of sleep spindles, where you've got these, 12 to 14 hertz waves that happen quickly and last about half a second. You also have what are called K-complex waves that look a little bit like the deeper sleep, where you get all of a sudden a sharp, high amplitude wave, negative and positive, that looks like kind of these bigger waves, but tacked into stage 2, almost as if stage 2 kind of gives us this middle ground, which again should make sense. It's not like there's this, which is completely different than this, which is completely different. It's like, no, this is stage 1, REM. When you get to the stage 2, things are a little different. You see these components that aren't the same thing. And as you go deeper, you see these others. Stage 3 and 4 together constitute what's called slow wave sleep. You get these long, slow waves, lots of parts of the brain kind of in synchrony, where you get this, slow waves, but very large. Well, REM is characterized by rapid eye movement and is usually happening during this particular stage of sleep. It's also called paradoxical because in some ways it's deep sleep, but in other ways it's not. But the EEG waves are irregular, low voltage, and fast. Postural muscles of the body are more relaxed than the other stages. When we get to, and that shouldn't be too surprising because in a lot of ways we think about it, when we have these kind of active dreams, we want the body to be relaxed because you don't want to act these out, right? We've got the pawns controlling that. So falling asleep, people go through 1, 2, 3, and 4, these stages, and then they start to cycle back through the stages from 4 to 3 to 2, and then REM. These cycles last about every 90 minutes, but there's variations between people and throughout the night. And you can actually, in a lot of ways, record yourself. There are apps for smartwatches that are able to kind of track your sleep by looking at how much you're moving to get an idea of your stages of sleep. Stage 3 and 4 predominate early in the night, and the length of the stage decreases as the night progresses. You have less of them throughout the night, where REM is greater later in the night, and the length increases. And what we're going to look at is what's happening to the brain and body during these different times. Why is it important to get these different types of sleep? Because of what's happening under these circumstances. So we see that different things are happening. Also, you don't have to be in REM to dream. You can dream in other stages. Though much of what we know about this comes from people going to sleep labs, going to sleep, and being woken up periodically throughout the night, and having the researchers say, were you dreaming? What were you dreaming about? We can usually, even for those of us who don't remember our dreams, we can usually remember right about when we wake up. So if they wake you up and say, what are you dreaming about? You can usually access that. And that's how we know a lot of things about the content of dreams, because of these things. We look at the mechanisms involved, and here we're looking at the reticular formation that extends through the brainstem and the midbrain, and extends connections throughout the forebrain that maintain awakeness. So you basically, the pons and the medulla are these deep structures in the brainstem that are extremely involved in our overall function, our basically overall alertness, awakeness. Damage to these areas are often fatal, because if you knock out some neurons in the reticular formation, your brain can't wake up anymore. We look at what's called the pontomesencephalon. This is part of the midbrain, but also part of part of the pons, this kind of connection here, which extends to all sorts of areas in the brain, which release hormones that keep it, or neurotransmitters that keep us awake. They produce excitatory effects all through the cortex. And if you stimulate someone who's sleeping here, they will wake up immediately. And if someone's awake, stimulating the pontomesencephalon will increase their alertness. We also have the locus coeruleus we can see here. It's a small structure in the pons, and these axons release norepinephrine. If we recall our ducts of the synapse and neurotransmitters, this is involved in arousing various areas of the cortex and increasing wakefulness. And we know that it's part of our, sympathetic, parasympathetic systems, or autonomic nervous system. Usually this area is dormant while sleeping. The hypothalamus also contains neurons that release histamine. Now, histamine is something that's all throughout our bodies and has interesting different functions. In our bodies, it tends to cause us inflammation. For instance, drugs that people take called antihistamines reduce that. So if you have an allergic reaction, an antihistamine will generally reduce that the intensity of that reaction. In the brain, however, histamine produces excitatory effects. And so when people take an antihistamine, it can cause sleepiness. But what's interesting is the old antihistamines, like Benadryl, or the active ingredient in Benadryl, is the same thing that's in a lot of over-the-counter sleeping pills, because this old antihistamine can cross the blood-brain barrier. And often it's cheaper to buy a generic version of Benadryl, the allergy medicine, than it is to buy a sleeping pill, even though it's the exact same drug, often the exact same amount. Well, the newer antihistamines can't cross the blood-brain barrier in most cases, and that's why they're able to relieve people of, maybe hay fever symptoms without causing sleepiness. We also have orexin. Orexin is involved in staying awake, and it stimulates the acetylcholine-releasing cells in the basal forebrain, the basal forebrain being just anterior to the hypothalamus, right? Basal forebrain being, here's our neural tube of the developing human, curves around, and it's that part right here. A lot of signal running through here in terms of wakefulness and arousal. Orexin is extremely important in maintaining wakefulness, and without it, fall asleep. We look at these cells of the basal forebrain, where we also have this, GABA release. GABA is inhibitory, and it's essential for sleep. You need to have this release of GABA in order to sleep. So you've got these different things in this area that are actually sending the signal for, all right, is it acetylcholine time to wake up? Is it GABA time to sleep? We know GABA is the brain's primary inhibitory neurotransmitter. And a lot of drugs that people take to sleep by prescription influence GABA. and boost GABA. One of the side effects is it also creates a boost of dopamine, a downstream boost, which creates this learning and addiction cycle. Some of those drugs like Valium and Xanax have been shown to be extremely addictive. Functions of the inhibitory GABA are important for decreasing temperature and metabolic rate and decreasing neural stimulation. But again, this should make a lot of sense to us. You boost GABA, you slow things down. GABA also works with alcohol. This is from Criswell and Breeze, 2005, where without alcohol, ETOH is shorthand for alcohol, this is what our typical GABA receptors look like. When you and glutamate. When you boost it, you increase the function of your GABA signal, and this creates this downstream effect that decreases the effect of your glutamate. So you basically, alcohol causes drowsiness. People sometimes act wacky and get hyper because a lot of alcohol has a lot of sugar in it, which can make people feel a little energy. But the ETOH, the GABA, the alcohol can reduce inhibition. So early on, people might start to act strange, but after a time, it increases neural inhibition and sleepiness. However, severely increased neural inhibition over time can lead to cell death. And when you get the glutamate burst later, that can also lead to cell death. But most people who've drank alcohol are aware that it's not particularly good for you, especially in larger amounts. And any study that says red wine's good for you, I tend to be skeptical of studies that say three things. If the study says red wine or alcohol or chocolate or caffeine are good for you, I tend to be a little skeptical. I'm not saying they're wrong. I'm saying we want to believe those things, and the media tends to report on things like that really excitedly. Like, oh, look, drink a lot of wine, eat a lot of chocolate, and then have caffeine in the morning to live forever. And it's like, no. All right, so sleepwalkers are interesting because sleep can be local in the brain, which means it can be in a particular location, and other parts of the brain can be awake. Lucid dreaming is an example of this. So some people have this ability to access kind of when they're dreaming and take control of it. Sleep paralysis is when the pons remains in REM, but other brain areas wake up. It causes the inability to move. Many people have experienced sleep paralysis. It's so prevalent that globally there are various cultures have a term for like the sleep paralysis, a demon, where what happens during sleep paralysis is an out-of-sequence REM state, where, again, part of your brain's in REM and part's not. So what are some of the symptoms of sleep paralysis? You feel like you can't breathe. And you feel like you can't move, both of which you can't do because your pons is still keeping you from moving and breathing for you. So also what happens, though, is you have these hallucinations that are very common of some kind of demon. So what you end up with, you tie all these pieces together. Your brain's not really awake. You kind of feel like you're hallucinating. You feel like you can't breathe. And this is interpreted as pressure on your chest, basically suffocation and panic. And yet you can't do anything because your body's paralyzed. Oftentimes when people learn that sleep paralysis is a natural process and that you can just go back to sleep and you'll be okay, it can often help, right? So it can often help. We look at what happens during REM. The pons increases activity. It's actually what triggers the onset of REM. The pons is heavily involved in sleep and is going to send a lot of signals through. And you've got these decreased activity everywhere else. So you've got decreased activity in your visual cortex because you're not getting any input, but you can still have, memories. You can have random things being produced. If you've ever done like sensory deprivation, you go into a completely dark room, no sound, in a tank with water that's heated to skin temperature, so you've got no sensory input, your brain will fairly rapidly start to do stuff on its own. We also look at these PGO waves. So the PGO is basically pons, geniculo, and occipital. So you're running from the pons to the G, which if you remember the lateral geniculate nucleus in terms of the signal from the visual system, from the retina to the LGN to the occipital lobe. Here you've got pons, LGN, occipital lobe. And what happens is, if you don't get enough REM, weird things happen. So if you don't get enough sleep, you can die. Particularly if you don't sleep for a long period of time. It's very hard to stay awake for a long period of time, but there are rare cases. So what happens is this: if you don't sleep, let's say you stay up all night because you're partying or you've got to school the next day or something, you've got to just kind of keep going and you're like, I'm fine. Well, the longer you stay awake, the more you're going to have these PGO waves start to creep into being awake. And you might start to hallucinate, which means you're driving along and all of a sudden you think you see a dog run into the street and you whip the wheel and crash your car, right? That's part of the danger of sleep deprivation is, these wakeful hallucinations. Now, generally you have to be severely sleep deprived to have this, but again, getting regular sleep, regular body rhythms is important, and people don't often take care of those things. So, REM is regulated by serotonin and acetylcholine, and drugs that stimulate acetylcholine receptors quickly move people into REM. Serotonin interrupts REM. One of the side effects of certain drugs given for depression that affect serotonin is difficulty sleeping, insomnia, and it interrupts part of this process. Insomnia can be caused by a lot of things, but a great deal of it is our modern culture, right? So you've got these lights, you've got people wanting to stay up, you've got noise, stress. Anybody feel stress? Exams, tests, work. A lot of things can cause insomnia. Late caffeine. A lot of people will have like a cutoff. Stop drinking caffeine after noon because the half-life of caffeine is a long time and it can make it difficult to sleep. Well, it can also be caused by certain medical conditions. We think of depression causing more sleep, but can also cause insomnia. How it just can disrupt sleep. Sleeping pills, dependence, alcohol, these can change circadian rhythms and create insomnia in the long run. It's extremely common, right? Most people have experienced it at some point. We also have sleep apnea, which is interesting. Not quite a traditional sleep disorder, but it can have cognitive effects, and that's what we care about here. So, sleep apnea is the airway closing during sleep. Many people will have their airway close over the course of the night multiple times, but for people with sleep apnea, it happens a lot, and it happens to the point where they are waking up constantly throughout the night. They're never getting deep, restful sleep because their airway cuts off and their body has to wake them up in order to open the airway. And these numbers are going up related to, for instance, obesity. So not all sleep apnea is caused by obesity. A lot of young people who have sleep apnea are in great shape, good health, and it's genetic. But it can also be related to obesity, and the numbers in the U.S. have doubled over the past couple of decades because of this. Now, the consequences are oxygen deprivation, which means during the day people are extra tired, they're not functioning well, and it can cause cognitive deficits. It can even kill you at a younger age by causing extreme physiological stress. Suffocation is not good for you. Narcolepsy is another disorder. This is the one where people just fall asleep during the day. It can be gradual or happen all at once. And they have, before it happens, what's called cataplexy, which is muscle weakness that can be triggered by strong emotions. You ever seen the fainting goats where they can scare the goats and they all just kind of faint. That's cataplexy. Sleep paralysis is common in narcolepsy, and also hypnagogic hallucinations. Now, we have hypnagogic hallucinations. If you don't have narcolepsy, you have this when you're falling asleep, right? And you start to have this hypnagogic sleep where you start to dream a little bit, but you're not quite asleep. A lot of times people see things, they think they see spirits or something. This is internally generated, but it's hypnagogic. You're not quite asleep, but you're getting some of this early, kind of dreamlike experiences. Well, narcolepsy is familial. It does seem to be genetic, but the gene hasn't been identified. And what happens is hypothalamic cells don't produce orexin. And if your hypothalamic cells don't produce orexin, you can't stay awake. Orexin stimulates the acetylcholine-releasing cells that stimulate neurons responsible for wakefulness and arousal. And so if your hypothalamus doesn't produce orexin, the problem is narcolepsy. The consequence is narcolepsy. And the treatment for narcolepsy is usually stimulants, which is not like a great treatment. It's like, here's some amphetamines, stay awake, but it's still not quite a perfect treatment. It doesn't quite do what orexin does. Night terrors are interesting. Someone may wake up screaming, but they're not dreaming. So there's just something else happening in part of their sleep process that's causing these. Sleepwalking can run in families. It usually occurs in stage 3 or 4, and there's some interesting things here. Should you wake a sleepwalker? Sure. There's this myth. Never wake a sleepwalker. Why? Be kind. If you can get the person back to bed, sure. But think about it. Like if someone's walking down the stairs, don't yell, "What are you doing?" They wake up, they fall down the stairs. But, if somebody's wandering around, it's okay to wake them up and bring them back to sleep. More common in children. And usually in deeper sleep. These people aren't dreaming. Another condition similar to sleepwalking is called sexsomnia, where people will literally engage in sex while sleeping and not remember it. This is dramatized, in kind of hospital shows where, someone doesn't know why they keep having sex or doesn't have memory of it. It's not common. But you do see cases of people behaving strangely, abnormally during sleep. Walking outside in your underwear to build a snowman, in this case. Well, why do we sleep? And here's the last big component of sleep, right? It is what's happening, why do we sleep, how are we dreaming? Well, the historical view has been this energy conservation, and we think about restoration. Well, what about memory? Let's look at these claims and see what there is. So, originally it was believed we sleep to conserve energy. Yeah, but not really. So you think about the amount of calories you save. Your body temperature drops a couple degrees Celsius. Okay, but that's not saving a ton of energy. Muscle activity decreases. But again, if you ate a couple hundred more calories, it wouldn't be an issue. Then you could just be up all the time. It's not really doing a lot to do this. So that's fine, but it doesn't really do it. We do know that, mammals that hibernate, do go through this. It does conserve energy, but it still burns a lot. You think about how animals bulk up before hibernation in order to put on fat to kind of maintain them through this. So they tend to do it because, food's scarce, and it does conserve more energy than wandering around in winter conditions trying to stay warm and hunt when there's no food. So they're, but that's more of like a behavioral and environmental effect. Animals sleep more if they are hunters than if they are prey in most cases, right? If you're not worried that something's going to come and eat you, you're just going to lie down on the Serengeti and take a nap whenever you feel like it, right? If you're, a herd animal that's worried about being attacked, then you have to be on guard. You're going to sleep less. Also, those prey animals tend to be herbivores, which means they need to eat a lot. eat all day, eat constantly in order to get enough nutrients to survive. Other interesting things happen. For instance, dolphins tend to sleep half brain on, half brain off in order to breathe, because they have to kind of be aware to breathe. Mother dolphins, when their babies are born, both the mother and baby won't sleep for 2 weeks about. And the reason is they need to teach the baby, and to stay awake with the baby, to teach the baby how to breathe. Because remember, they have to surface to breathe. And if you breathe underwater, you breathe water and you die. So the mother dolphin is teaching the baby how to do this process. Migratory birds will often sleep with half of their brain off at a time in order to maintain, flight. Now, memory is interesting because sleeping is really good for learning, right? Now, there's a myth about consolidation, like right before bed, your brain consolidates. You're consolidating throughout the day. There's other things happening that enhance this. But we do know that sleep is good for learning, and that the brain is going to replay those things while you sleep. For instance, your memory centers during learning were like those during sleep. So if you have learned something, your brain is going to do those things while you're sleeping. The brain will strengthen some synapses and weaken others. And this is very important. Your brain needs to get rid of stuff. You remember things from today that you don't remember from yesterday. What did you do this morning? What did you do 5 hours ago? What did you do yesterday, the day before? After breakfast, an hour after breakfast, 3 days ago, what were you doing? You're like, what? So we put a bunch of stuff in our brains throughout the day. When we sleep, this weakening of synapses is extremely important. You're maintaining synapses that are useless, right? You can picture yourself walking through the aisles of some store or, in class or whatever. It doesn't help you. It doesn't do anything for you. You need to get rid of that stuff, right? And sleep is helping you do that. It's clearing out stuff. We also sleep spindles associated with learning and correlated with nonverbal IQ. We know that we spend about a third of our lives asleep, and about a fifth of that time is spent in REM. The more you sleep, the more REM you get for humans and other animals. And it's not entirely sure what REM does. It seems to be associated with memory learning, but some of the deeper stages are too. And it's so difficult to understand. There was this suggestion from Maurice in 1998 that REM is to shake the eyeballs back and forth, right? How your eyes move when you're sleeping, in order to provide sufficient oxygen to the corneas. Because it's important you get oxygen. So it's kind of like working out your eyes while you sleep in order to make sure that your corneas are oxygenated and healthy. It may sound odd, but it's not entirely clear. We're starting to understand why we sleep, but not be clear on all the different stages. Now, a big one is clearing out toxins. This is from a 2013 piece where it was shown that when we sleep, you get an increase in what's called interstitial space, which is this fluid exchange between the cells and cerebrospinal fluid. So basically, the fluid is kind of leaving the cells and taking with it things like beta amyloid. Beta amyloid is one of those things highly present in Alzheimer's disease. And when we sleep, we clear out this stuff. Neurotoxins build up in our brains throughout the day. Sleeping allows this mixing of the fluids, which gets rid of these toxins, damaged proteins, and other things that might be bad for us. So one of the clear, important functions of sleep is brain health. Getting rid of toxins. Mixing of the fluids from the cells with the cerebrospinal fluid. Cerebrospinal fluid runs all the way down from your brain to your spine, mixes with your blood, and you eventually excrete this stuff. Learning. This is a 2014 piece. Basically, when we sleep, it is clear that we increase our dendritic spines, basically growing new synapses. So the more we sleep, the better we are with a task. They took the rat, they taught it to run on the spinning rat, Some of the rats they kept awake and some of them they let sleep. The ones that slept did better the next time they tested them both. And when they looked at their brains later, they had more dendritic spines, which means they had reinforced this learning in their brains. So, memory enhancement, learning, brain health. Sleep is wildly important. You can't get rid of this stuff. The synapses you don't need. You can't strengthen the ones that you do. You have to sleep. And again, this is one of those things where we have this weird cultural, like, oh, it's so cool to not sleep. Oh, I only need 4 hours a night. That's not good for you. You're probably going to have some long-term consequences, including a increased likelihood of dementia. Now, what about dreaming? Here we see that research on dreaming is tough because people can't often remember what they dreamed. But what we can do is wake them up while they're dreaming. So we have a pretty decent idea of this, but why people dream is best described through probably the activation synthesis hypothesis, but the clinical anatomical hypothesis is useful too. And activation synthesis is this. You've got spontaneous activity in the brain. And eventually that part that ties it together, probably the thalamus, wakes up and says, okay, here's these different brain areas are doing something. I'm going to tie them together and tell a story. So we're telling a story about why these different brain areas are active. It doesn't know. The brain doesn't know why they're active. But this is what's active. So there must be a reason for it. And there's our dreams. Look at our common experiences. Anxiety dreams, like, oh, I didn't study for a test. Well, it's a common anxiety thing. Being weak, maybe trying to run and not be able to do it well because your body is weak. Your body is being basically, kept from moving by your pawns. The clinical anatomical hypothesis suggests that dreams are similar to thinking, but the situations are unusual. But in this case, yes, dreams begin with these stimuli that generated from within, but the stimulation is combined with recent memories and sense information. But we know the brain is getting little information from the sense organs. So these images are generated without constraints or interference. You are not you in dreams because all the parts that make you are not functioning. Let's see. Okay, let's look at the parts of our brain that are active in our dreams. Well, our frontal lobe is associated with rational thought. If your frontal lobe is asleep, dreams may be more likely to be irrational. Your parietal lobe, right, your physiological, finding vision and body sensations. People who have damage here don't have dreams. The occipital lobe, high activity in dreams. Also areas outside of the occipital lobe, giving us visual imagery. Visual imagery is the most intense. We tend to have dreams with visual imagery, but not as much dreams with sound or body sensations. The thalamus is likely tying these things together. And the hippocampus is giving us memory, though it may become inactive and not save the memories in the dreams. So you have maybe pulling out memories, but not really remembering our dreams. Our hypothalamus and brainstem are associated in REM, rhythm, sleep onset, and our amygdala is associated with emotion. And what this gives us is these experiences. Sun says our emotional experiences. But dreams can be very emotional. All right, so this method of studying the brain allows you to view attention changes in real time. Is it EEG, TMS, CT, or fMRI? And the answer is EEG. What is happening right now? fMRI tells you more about where. This method of studying the brain allows you to see approximately where processing is taking place. And that would be fMRI, where things are taking place. TMS allows you to change things. CT allows us to see the brain structure. And we do want to take a look and understand these things. But we talked about EEG today because that's how we know a lot about sleep. But we do want to remember all of our areas. This concludes our discussion about sleeping and dreaming, and these states of consciousness. I hope you enjoyed it, and I look forward to future discussions.